be brave if you're brave. I'll be brave, but only if you're brave. And it could be just you and me. We'll be family. Just wait and see. So I will fight if you'll fight. Yeah, I will fight, but only if you'll fight. Oh, we can make it through this like sailors in a tempest. Like sailors in a tempest together. And it could be just you and me. We'll be family. Just wait and see.
Good evening, everyone. Welcome to tonight's Lung Cancer Living Room. Welcome to everyone in the room, all of those joining online via YouTube or Facebook Live or coming back to watch us post live. Welcome to all of you too. We are so excited to have everyone here tonight, and of course, our guest speaker, who really needs no introduction.
His Excellence.
His Excellence, Dr. David Gandara from UC Davis Comprehensive Cancer Center. You guys have either seen him here in the room, watched him on video, or heard us talk about him at almost every living room. He comes up in some form or fashion. So we're really excited to have him here tonight to talk to you all about personalized cancer circa 2019. Where are we today, and what does personalized medicine look like today compared to how it has looked like in the past, and where's it going? So what I'm going to do before we open up and do the last hour and change, educating everyone on tonight's topic, I'm going to ask Dr. Gandara to tell us a little bit about himself. As you guys know, I used to spend a lot of time like this and a piece of paper reading our amazing speakers' bios and whatnot. And it just didn't seem very personal to me. So about a year and a half ago or so, we started just asking our speakers to introduce themselves. Tell us, why they're here, why lung cancer matters to you, why you chose lung cancer as your sort of spot to lay your head and do your good work. So you want to give us a little bit
and then I'll add whatever he forgets.
Yeah.
I think I'd like to start by saying the reason that I'm a lung cancer oncologist is because of this room. I mean, it's incredibly inspiring, and not just to me, but I'm sure to all the other speakers that come here to hear these stories. I could speak for 3 days just based on your stories. But I grew up in Texas. I went to the University of Texas in Austin, and then medical school in the University of Texas Medical Branch in Galveston. And then I came to the West Coast, and I'm still here. And met my wife here, and I've been at UC Davis. She's a 4th generation Northern California
And an angel.
girl. She is.
Yeah.
Some would say she's an angel to put up with me.
That's right. That's what I was referring to.
But I've been at UC Davis for 25 years. I was in the military. As a medical officer, did my training at Letterman Hospital on the Presidio, which is now a national park, you may know. But at the time I did my training there, it was the biggest hospital, biggest training center in Northern California. We had more fellows, more oncologists, hematologists than Stanford or UCSF. So it was quite the place in its day. And then after that, I went to UC Davis, where I still am. So I was for 20 years the Associate Director of Clinical Research. So everything that had to do with patients. And I helped take UC Davis from a brand new cancer center to what's called an NCI-designated cancer center, to an NCI-designated comprehensive cancer center, and the fastest trajectory in the history of the National Cancer Institute. So we're very proud of that. I've since given up that role to, many of you know Karen Kelly, who's with me at UC Davis. She's now in that role. But I remain the Director of Thoracic Oncology. I have a brand new 6-year grant from the National Cancer Institute for $3 million to develop new treatments. So I don't plan on going anywhere anytime soon. I love to teach. I think this is probably my, maybe 5th time over the years. How long have you been doing the living room?
You've come way more than
10 years. We've been doing
10
the living room for almost 10 years.
Well,
you come at least once a year.
Yeah.
all right. So at any rate, I love coming here, and I always tell everyone that education is bi-directional. And it doesn't matter whether you're speaking to a medical student or a fellow in hematology oncology or to patients and their caregivers. I learn.
I think it's so important, and I think when we're looking at research and how important it is, and you guys know we talk about it all the time, we're going to talk a little bit about where research has brought us today and maybe where it's going to take us in the future. But where I want to start, Dr. Gandara's done a great job talking a little bit about personalized medicine and what does that mean, and about molecular fingerprint and how nobody's cancer is the same as the person sitting next to them or the person across the room. So I think it's sort of apropos to open this conversation with the molecular testing component, or biomarker testing, or whatever, genomic profiling, or one of the many things that it's continually called. What does that mean? What does that look like? Specifically, those 8 oncogenes, and what beyond that?
Right. So I think the introduction for this topic would be to say that 10 years ago or 20 years ago, we had research. And then it took years and years before that research came into clinical practice. These days, that has been turned on its head. And what was research yesterday is clinical practice today. And that's where we are for molecular testing in cancer, and in particular for lung cancer, non-small cell lung cancer specifically. We know that it's an incredibly complex cancer in terms of mutations. So that molecular fingerprint is more complicated than most other cancers. We started out knowing that one of these mutations, and it was the EGFR, which quite a few of you have, could predict who benefits most from drugs against that target, against EGFR. So one thing I'll have to say is almost everyone in this room use brand names. I can't use brand names. I have to give the generic names. So you may not know what the drugs are when I talk about them, but you can look on Google and it will tell you. So those initial drugs, gefitinib and erlotinib, changed the life of cancer patients around the world that had that kind of mutation. And now we have not even second generation, but what we call third generation agents, like osimertinib, otherwise known as
Tagrisso, some of you said it earlier.
But anyway, that's where we started. And the point Danielle is making is that now, today, in 2019, we have 8 of these markers that will predict who will benefit most from that drug against that target. And you heard about one of these earlier, the Loxo drug, which is against one of these called RET. Highly active, and we have just many examples of this. What that means is if you have non-small cell lung cancer, advanced stage, typically stage 4, and especially if it's an adenocarcinoma, in 2019, your cancer needs to be tested for all of these 8. I saw a patient this past week, otherwise very, very good oncologist, and she said, well, we had to do such and such because the patient didn't have EGFR, they didn't have ALK, and they didn't have ROS1 in their cancer. And so my response, in part, was, what about the rest of them? And the reason I said that in this patient is she is a never smoker. That should set off a bell that this patient is highly likely to have one of the 8 oncogenes because they're more common in never smokers. So that's one of the things that I did when I saw her last week, is realizing that she needs new treatment right away. And even if there was residual tissue, it would take some time to do the testing. I drew blood on her to send it off for a panel that within 7 days will give us the results. And I'll just mention the name. It's Gardant, the Gardant blood test. And again, Barbara will appreciate this. Their turnaround time is 7 days. That's 7 total days. Every other test tells you about working days. In other words, they don't count the weekend. You could be in Singapore,
Right.
or you could be in Abu Dhabi. You will get this test back in an average time of 7 days. So in 7 days, we'll know about the rest of these oncogenes in this patient. So that's,
So,
yeah.
so we've talked about the 8 and sort of what, when I talk to patients, I talk to them about like just sort of this baseline lung specific testing versus like a comprehensive test. What are the benefits to, like at minimum, these patients should be getting tested for the 8, right? But what about and when does it make sense to do a more comprehensive testing?
Right. So this is debatable. I should mention actually though that we're about to have number 9, G12C KRAS mutation. Because of some
30%
new
of lung
drugs like
cancer.
absolutely hot off the press.
Yeah.
Hot off the press within the last month.
Yeah.
So it's going to be number 9. I can almost
Yeah.
guarantee you that.
Huge.
So the issue about testing is that 15 years ago, when EGFR was discovered, we tested just for EGFR. Or some people said, well, test for KRAS because we already have that test around. Even though we didn't have anything to do for KRAS, they said, if the tumor is KRAS positive, it's not going to be EGFR positive and you don't have to do any more. That was wrong. But in the meantime, all of these other abnormalities, which we've heard about, some of them in the room today, ALK and so forth, they all came about. So in 2019, some oncologists or pathologists who direct the laboratory design for the testing will test for one, then test for the next, then test for the next. That takes forever. So what you want to do is test for all of them at the same time. So there are other platforms that we call sequential, right? You test for one, then the next, the next. There are exclusion tests that will test for 5 or 6, but don't test for all of them, and also don't allow you to assess other abnormalities such as the tumor mutation burden. And then there's what we would call next generation sequencing, where literally hundreds of genes are tested. And that gives you all the 8, plus a lot of other things, which some of them are meaningful, some of them are not, but you need them to give other information such as the TMB. I know this is complicated, but what this means is in 2019, if you can get next generation sequencing done on your cancer, that's what you should do. There was a really nice recent study looking at comparative effectiveness or cost effectiveness of these various ways: sequential, exclusion, next generation sequencing. And Dr. Pinnell here in the United States, Cleveland Clinic, showed in a modeling study that you actually save money by doing the next generation sequencing because you test for all of them at the same time. One test, boom, you've got the information. And then, as we'll come to in a minute, we can now do exactly the same thing in blood. Blood and tissue are a little different. Again, that's complicated, but we're about to do a new consensus paper for our ISLC where we're going to say blood is now moving up front And maybe you don't need to do tissue first, maybe you can do blood.
So I have a question, and I just thought about it as you were talking, and we've had these conversations at the living room before and obviously in other places. When we're talking about next generation sequencing, and I'll use another block, so like let's use NTRK. It's a pretty small percentage of the population, right?
So she's getting way out there. We're like circling Pluto.
Yeah, but here's where my question's gonna lie, because I'm trying to think of patient benefit from a research benefit, I guess, from what we know about the patient. Like, had Loxo had information on patients who had NTRK and could have filled their trials quicker, could we have gotten drug to patient faster? Do you know what I'm saying? Like, if we had this genomic profile.
So the answer is yes.
Okay, thank you.
How many of
reason
you have heard about the Lung Master Protocol or Lung Map? Have you heard about it? Some people have. Okay. So we developed this. Actually, my colleagues in crime, I guess I should say, Roy Herbst and Papa Dimitriopoulos.
Yeah.
And won an award from the Addario Foundation on team science. Because we got together with the National Cancer Institute, with the foundation of the NIH, with 20 pharmaceutical companies, with Foundation Medicine, which has a testing platform for molecular testing, and with the FDA to form this master protocol where if you're a patient that gets entered into it, we will test thousands of patients per year, thousands. We have 500 sites in the United States. And we will place the patient, as the studies are available, into a trial where you get a drug against your mutation. And if you don't match, we have several immunotherapy trials where we match you with what seems to be best for your cancer with immunotherapy. So both the genomic and the immunotherapy. So if there's a rare abnormality, an NTRK, a RET, then if we're testing thousands of patients a year in 500 sites, we will find the 2%ers. We'll find plenty of them to do the trials and show that the drug is either active or not, because part of our mission is also to rule out drugs that sound good. Maybe they work in breast cancer, or they work in hematologic malignancies, or they work in bladder cancer. And I use those three examples because there are drugs out there for those three cancers, same mutation. In lung cancer, they don't work. So we're about to initiate a confirmation trial for the FDA with the Loxo drug in RET-altered cancers. So we can do that faster than any company could do it because we're testing all of these patients. The very same is true for TRK, and we're going to have a TRK study as well.
Yeah. I'm honored to say that when Lung Map materialized via SWOG, Southwest Oncology Group.
Where Bonnie was our patient advocate.
I was, and I got to contribute a lot to that, and it was a great learning experience for me as well.
But I think, I guess
such an amazing.
The takeaway here is that this molecular testing, genomic profiling, biomarker testing, whatever you want to call it, is important for so many more reasons, right? The obvious, you would like to find a target where there is an approved drug for your cancer. However, if you don't, and Rebecca's a perfect example of that with her KRAS and whatever might be going on, right? That if it's not available now, maybe it's available down the pike, and it's good information to have in your back pocket for if and when this ever comes to fruition.
If I can just add one more.
Please.
So when I said Lung Map is available in 500 institutions, almost all the institutions in the Bay Area have it active. Kaiser has it active, Sutter, Dignity, most of the sites have Lung Map active. So I realize it may not be for all of you, but it could be for some of you. Yeah. So keep it in mind.
So I'm going to try not to sound like the fool when I ask this question, but going back to small cell for just a second. You touched on immunotherapy, and I do want to dive into some immunotherapy conversation before the time's up. But there was a DL8?
DLL3.
L3. Thank you.
Yes, don't, he's our other
I know,
consultant.
he is. What's going on there?
So that's a long story.
Yeah.
But this is a drug that has a wonderful rationale for it. It's a specific inhibitor of this abnormality, which is very high expression in small cell lung cancer. And the early data seemed very positive. But that's the reason sometimes that we really need to do phase 3 trials, where we compare it to whatever's the standard of care. Because over and over again, we find out that early data from a small study may not play out the same way in a bigger trial. So I know patients don't want to be randomized. Most often now, patients are not randomized to only a placebo. It may be standard of care, which it could be chemotherapy, could be immunotherapy, could be a variety of things, plus a placebo versus the new drug plus the standard of care. But in this case, the drug didn't pan out well, and it ended up having a lot of side effects.
Did it?
So it's not probably totally gone yet, but it's what's called a drug antibody conjugate. That means you have something that goes into the tumor because the tumor is positive for it, but you hitch on something that's a chemotherapy that once it's in the cell will get released and kill the cancer cell. And those linkers are very complicated to make. And part of the problem with this is the linker didn't make it go into the cancer cell completely. So the concept is totally valid. There are other drugs against this target still being developed now, but I'm not sure this drug's ever going to make it.
But this, I guess.
But you're right, it would have been the first targeted.
Yeah, and it kind of points back to the importance of this testing in all tumor types at some point on some level, right? Whether it's a non-small cell or a small cell, because without the testing, we wouldn't even know it was there.
That's true.
An initial testing.
Amy,
Can I throw in one more thing?
Yes, no.
So the other thing is that patients often, and I get this all the time, patients come in, they've read, and they come in and say, Dr. Gandara, I just want to let you know, I don't want a phase 1 trial. I only want a phase 3, or I only want a phase 2 or 3. And I'll say, why? And they'll say, well, I heard that these phase 1 trials are always negative and you never get anywhere. Every one of these new drugs we've talked about tonight was highly active in phase 1. Phase 1 trials today are not your grandfather's phase 1 trials. And there have been several publications showing that even people who go on a phase 1 trial with cancer have a much better survival than patients who get treated with whatever was the next available thing that the oncologists could have used. So you should know everything about the trial. You should know the risks, you should know the benefits, but don't be scared if they say it's a phase 1 trial, if the rationale seems good, because it may be a home run for you.
Oh, I'll be brave if you're brave. I'll be brave, but only if you're brave. And it could be just you and me. We'll be family. Just wait and see. So I will fight if you'll fight. Yeah, I will fight, but only if you'll fight. Oh, we can make it through this like sailors in a tempest. Like sailors in a tempest together. And it could be just you and me. We'll be family. Just wait and see.